see you today. Thank you for joining us today for our wHolistic, for our Understanding Parkinson's Through Neuropathology talk. Did everybody like that new intro? Yeah, kind of fun, up in the game here. So welcome everybody. So we are going to get started. We have Dr. Vinters and Dr. Subramanian. I'm going to pass it off to her. Hi, Dr. Subramanian.
Hi, how are you?
Great.
Thanks for joining, everyone. It's great to see everyone out here. I'm still warming up to this music. I'm not so sure quite yet, but I think it'll grow on me probably. I'm a bit of a creature of habit, but anyway, welcome everyone to our physically distanced but socially connected here virtually through our PMD Alliance group here. And with a lot of the questions that have been coming up recently have been asking about more and more questions around what happened with brain donation, what do we see under microscopes, and it really led me to be inspired to reach out to a good friend of mine and a close colleague who works at UCLA with me. He's really fabulous. He's a professor here at UCLA in pathology. He has the Daljit and Elaine Sarkaria Chair in Diagnostic Medicine as well. So it's very rare that a neuropathologist gets all of these honors and accolades, but Harry Vinters is really pretty spectacular. So he did not hesitate to participate because he actually, even though he works behind a microscope a lot of the times, he really enjoys talking about patient care and learning about patients and wanting to assist, in continuing to fight the fight against Parkinson's. So he was, happy to do this, and I actually ended up getting to see him yesterday in preparation. It was a good excuse to meet up, and just always a pleasure to see you, Harry, and thank you for joining us today. Maybe I'll let you introduce yourself a little bit. Harry is a fellow Canadian who trained in Canada. We went to the same medical school, and he can teach you a little bit about his journey because it actually started with wanting to be a neurologist for a little while, and then he grew into wanting to be a pathologist, and he can share a little bit with us, and also, any other key tidbits that he wants to share. I just learned that Harry is such a cool person that he is the godfather for 5 people's children. So he has 5 godchildren, which speaks volumes. I think I might have him become the godfather of one of my children as well. We'll see by the end of this. But anyway, thanks Harry for joining us, and I'll let you teach us a little bit about your journey, and then we'll get into your talk.
Great. Thanks, Indu. It's my pleasure to be here. As Indu has indicated, we're both graduates of the University of Toronto. We're both Canadians. I have 2 passports. Indu, I presume, has 2 passports as well, maybe even more. Indu is right. I started training in internal medicine and neurology. And then what really interested me in neurology was actually a rotation I did in neuropathology. So I asked my program director, who was kind of a legendary figure in Canadian neurology, Henry Barnett, who passed away within the last couple of years, well into his 90s. I asked him if I could do a full year of neuropathology, and he agreed to that because he was very neuropathology oriented. And at the end of that year, I decided to switch careers to neuropathology, and have done that. I actually, it's just pure serendipity that I'm at UCLA. I came down to do, after I finished my specialty training in neuropathology in London, Ontario, not London, England, London, Ontario, which was a very good program. I went down to do research at the University of Iowa. And the day I arrived at the University of Iowa, my preceptor there had just accepted the chair of pathology at UCLA. And since I had my own funding, he asked if I wanted to move with him to UCLA. So I did 2 years of research here, which was going along quite well. And at the end of the 2 years, nearing the end of my postdoctoral fellowship, he called me into his office and said, would you be interested in coming on faculty here? And I was, and went back to Canada because I was on a J1 visa, which didn't allow me to stay in the USA. Worked in Canada for 1.5 years and then moved back to UCLA in 1985. And I really enjoyed being here. I really enjoy working with the movement disorders people. Indu, Dr. Portera Kaliu, Yvette Bordelon, Jeff Bronstein are all good friends and colleagues. And Indu especially is very, very helpful when we do our medical student teaching every year, and puts on a very good demonstration of the neurologic examination and where it leads you for the medical students. This year that was modified a little bit, obviously, because of COVID-19. We did we did video presentations, but we'll be hopefully going back to in-person teaching next year. So that's about it. Indu jokes about my 5 godchildren. When they were little kids, that was actually quite expensive because they all expected gifts on their birthdays and at holiday times. They're now thankfully adults, and it's interesting to see how they've all developed. One of them is actually quite a senior investigator at the Centers for Disease Control. So I talked to him actually last week, and he was telling me about all the ins and outs of COVID-19 testing and all the problems the CDC has gone through. So very interesting from that point of view. And what I plan to do today is just give you a little summary of what happens with brain donation, with the autopsy, and how valuable it's been in teaching us about pathophysiology and neurologic diseases in general, but especially in Parkinson's disease. I should warn you that we've had a couple of problems with what's called black screen with Zoom, but it tends not to happen until about 40 minutes or 45 minutes into a talk. And I think my talk's going to be about a half an hour, 35 minutes, so we should be safe. If that happens, we're told to just log out and then log back in and continue from there. So we'll finish with that.
Perfect. And Harry, maybe you could also teach us a little bit, because this has been a really enjoyable thing where we learn a lot, what brain cutting is. I don't know if you're going to mention it in your talk, but I think it's been a really nice marriage for us as clinicians to be able to come to, effectively, where we see the brain sometimes of our patients that we've taken care of and had relationships for so long with, who may have donated the brain, and we stand near, Harry or a colleague and are able to actually see the brain in front of us and learn a lot about this field and kind of keep getting inspired. So I think this will be helpful.
Great. Should I go ahead?
Yeah, go for it.
Okay, great. Sorry. Excellent. Well, again, it's my great pleasure to do this. I enjoy interacting with clinicians and the movement disorders people, and I've been interested in degenerative diseases of the brain, and I'll say quite a bit about that and how we study those diseases using histopathology. And Dr. Subramanian and I were talking about this yesterday, that autopsy studies have their limitations, and they certainly do, because we're looking at autopsy at brain specimens from individuals who've had disease for many years, for quite a long time, and we're looking at the end stage of that disease. But it's a very important study, because those autopsy studies have really shown the pathways for biochemical and molecular analyses in degenerative diseases, including Parkinson's disease, and I'll say specifically many words about that, and also in Alzheimer's disease. These are now often referred to as diseases of protein folding, or misfolding diseases, because the fundamental abnormality in these diseases seems to be protein folding abnormalities. Different proteins, obviously, in Alzheimer's disease than in Parkinson's disease, than in other neurodegenerative diseases, but it is quite a common theme. So why do an autopsy? I'll tell you eventually what happens in an autopsy, but why do an autopsy? We're in an era when there is incredible technology that can help characterize a person's disease while they are alive. In the case of brain diseases, we have MRI, we have positron emission tomography, structural imaging CAT scanning, which was actually based on something that was suggested by a UCLA professor, Bill Oldendorf, who I had the pleasure to actually interact with when I first came to UCLA. Quite an amazing man, should have gotten the Nobel Prize for it. But imaging is very powerful. There are many powerful biochemical and molecular techniques that can be done in a living patient to try to characterize their disease. And actually the neurologic examination is extremely important. I always say that the neurologic examination is one of the most important biomarkers for a neurologic disease, because really tracking the progression of a clinical disease by physical examination and analysis of signs and symptoms is extremely important. So the autopsy helps to confirm the clinical diagnosis. If the diagnosis was cancer, is there evidence of cancer? If it was Alzheimer's disease, is there evidence of Alzheimer's disease? And if there was if the patient had Parkinson's disease, do they have characteristic features of Parkinson's disease in the brain? or do they have evidence of another disease that might give them Parkinsonian symptoms? That's an extremely important issue to address. The autopsy helps to establish the extent and severity of the disease. So even if the diagnosis was known clinically during life based on examination, imaging, and lab tests, it helps to establish the extent and how severe the disease was at the time of autopsy. There can be incidental or unexpected findings that may have contributed to disease severity or other symptoms. And very importantly, the autopsy provides materials to be used in research. For example, evidence from the pathology of response to treatment of a given disorder. And that can be true for both cancers and for neurodegenerative diseases. So I will show you how we examine and study the brain at the time of autopsy, and then that brain cutting exercise that Dr. Subramanian talked about. When we take material for research, we bank the materials in what's called a human biospecimen or brain and cerebrospinal fluid bank at UCLA. We have an endowed Alzheimer's Center, but the Alzheimer's Center also studies brain tissue and cerebrospinal fluid from individuals with other neurologic diseases than Alzheimer's disease. So we consider that we have a much more global mandate to bank those tissues. I always like to point out to audiences the similarities and the differences from traditional banks. So in a traditional bank, we place money in, we deposit money, we take money out, and the money earns interest. So the deposits into a biospecimen brain bank are from biopsies or autopsies, and most commonly autopsies. And those determine its value. So a bank's value is determined, I guess, by the assets under management, the assets in the bank. In a brain or biospecimen bank, the value of the intrinsic value of the brain bank is what its holdings are, and what those materials are and how well they have been characterized by the neuropathologist, which is me or one of the other neuropathologists here. The withdrawals are made, and they're generally made by researchers, or they're made by the neuropathologist who then forwards material to researchers. And we've distributed over the years that we've had the brain bank, which is now about 20 or 25 years, not only to materials to researchers at UCLA, but other researchers in California, and we've actually distributed materials throughout the world. So we know that, for example, autopsies are quite rarely done in Asia. We had a collaboration for a long time with a very productive Alzheimer's laboratory in Singapore, the Singapore National University, and submitted many specimens to them for their studies. So, and we've submitted materials to the United Kingdom, to Ireland, many other places, where they take advantage of the high quality material that we have from the very well characterized cases. So the question is, does the bank pay interest or dividends? Yes, it does, but those aren't in cash. The dividends and the interest are scientific advances and publications. So there have literally been hundreds of publications that have resulted and continue to result from materials that we've provided to basic researchers and translational researchers. So the dividends are not money, but they are scientific advances which are disseminated throughout the world and useful to other investigators. So can a brain bank fail, I guess is the question. There was a lot of talk about being too big to fail at the time of the 2008 financial crisis. They can certainly fail if they don't input material to the brain bank and if they don't distribute it to researchers as it should be distributed. So a brain bank can indeed fail, but the successful ones are an extremely valuable resource for researchers. So I'll show you a picture of what I call team neuropathology at UCLA. The 4 neuropathologists, the 3 in addition to myself, that's me, are Dr. William Young, Negar Kanlu, and Shino Magaki. Shino is with me in the room here. And the manager of the brain bank is at the back here, Kazu Williams, who's a jack of all trades and is extremely good at providing specimens to individuals who make use of that in their research, and is extremely helpful and is very good at doing many procedures as well. Now you'll notice that unfortunately we're not social distancing here. However, we did another photograph in which we are properly socially distanced. So These are the same people you saw in the prior photograph, but we're now on the steps of the David Geffen School of Medicine at UCLA. And again, this is Kazu Williams, who appropriately enough is at the front because he's a very valuable person in distributing brain bank materials and taking in requests. Our two histotechnologists are Nellie and Jennifer, and they're the ones who actually cut the sections. I'll be showing you examples of that allow us to make a diagnosis on a case to confirm the diagnosis of Parkinson's disease or another neurodegenerative disease. What actually happens during an autopsy? First of all, we will not do an autopsy unless we have a legal consent from the next of kin to do the autopsy. And if the next of kin wants to place restrictions on the autopsy, he or she is perfectly entitled to do that. So on many of the people with brain diseases on whom we do autopsy, we do what's called a restricted brain-only autopsy, in which we only remove the brain. We still get a lot of information from that autopsy, but we get a little bit more information if we are allowed to do a complete autopsy. And in fact, Dr. Magaki, my colleague, did a very important study some years ago reviewing the systemic findings in people with neurodegenerative diseases. And we now know that, for example, the protein that is misfolded in Parkinson's disease, which is alpha-synuclein, can actually be present in neurons, nerve cells, nerve fibers throughout the body, not just in the brain, although the clinical manifestations are usually in the brain. During the autopsy, the organs are removed from the body, weighed, and examined, including the brain. Most of the organs and tissues are sampled for histologic analysis, and I'll show you some histologic sections, and this happens at the time of the autopsy. Some material may be frozen for molecular, biochemical, and pharmacologic analysis and research. We treat the brain a little bit differently. So the materials for research we will sample from the brain right at the time of the autopsy, and the remainder of the brain is then fixed for about 1 to 2 weeks, and then is cut at the brain cutting sessions that Indu described. And then at the time of the brain cutting, the slices are examined, and I'll show you examples of brain slices, and digital images are made of the slices, and then they're sampled further for histology, and I'll show you examples of that. Now, when we look at the histologic specimens, the two techs that I mentioned to you, Nellie and Jennifer, cut histologic sections from those brains, and then depending on what the clinical suspicion is, we do a number of special stains and immunohistochemistries to fully characterize the disease. And we now have antibodies to those misfolded proteins in both Alzheimer's disease and Parkinson's disease. And those proteins and those antibodies have been developed certainly beginning in the 1980s when the misfolded proteins came to be identified. So we have very, very precise reagents that allow us to examine localization of those proteins in the brain, see where they are in the brain, and further characterize the disease. There are a number of trends in autopsy practice. Autopsies are rarely, sometimes never performed in community hospitals because they don't have the resources, and they will transfer the body to us to undertake the autopsy here. And we're perfectly happy to do that and help people out with that. They are more common in academic medical centers, but even in academic medical centers, the percent of autopsies that are done have decreased somewhat over the years, and I'll show you some graphs. But the absolute numbers may be increasing if a place becomes a regional autopsy center. So UCLA is really a regional autopsy center, and our numbers of autopsies are actually increasing every year. And we're going to do quite a few more autopsies this year than in previous years. But overall, autopsies are performed, and it's estimated less than 10% of deaths in the United States. Now, there are many different practices for autopsies. In some European medical centers, when a patient went into hospital, they pre-consented for their own autopsy. So literally everyone who passed away at that hospital had an autopsy, but here we require a separate consent. There are also financial reasons why autopsies are not done. So Medicare stopped paying for autopsies quite a long time ago, about 34 years ago. And the accrediting body for hospitals, called the Joint Commission on Accreditation of Hospitals, eliminated an autopsy requirement as a condition for accreditation, and even earlier than that in 1970, so about 50 years ago. So this removed the major financial and regulatory incentives for doing the autopsy. But the autopsy, if anything, is making a comeback in the last few years. This is a paper that came out about 12 years ago, just summarizing the trends in autopsies, autopsy rate. So autopsy rate is defined as the number of people who have an autopsy as the numerator over the total number of deaths in a given medical center. And you can see that in many of these world-renowned medical centers like Brigham and Women's Hospital in Boston, the Mayo Clinic, Yale New Haven Medical Center, the trend was definitely downward, but probably has stabilized over the last 5 or 10 years. And again, extremely important for us to gain information from the autopsy, both diagnostic information and material that can be provided to researchers. Why are autopsies in decline? Pathologists, frankly, are some of the individuals who are responsible for this. Many pathologists don't like doing autopsies, and that kind of is inherited by the pathology residents. We're very fortunate in that at UCLA we have a very dynamic chief of autopsy pathology, Greg Fishbein, who is a very, very good teacher using autopsy material and gives talks to clinicians regularly on the results of autopsy studies. But autopsy pathology, to be honest, is not as sexy as surgical pathology or molecular diagnostics. It's not a terribly remunerative part of pathology practice, but there are some exceptions to that, sometimes in medical legal autopsies. The other reason is that, well, there's the myth that everything is known about a given patient's disease based upon high-resolution antemortem imaging, neuroimaging, and biochemical studies. That's not exactly true. A lot is known about a patient's disease based on antemortem imaging, and there are now ligands for some of those misfolded proteins that can be used together with positron emission tomography to try to get a sense of the distribution of those abnormal proteins. But even those have led to some controversy as to exactly what is being imaged, how valuable is it, how important is it in following the patient. There is some concern that by clinicians that the autopsy will uncover findings that may lead to litigation. But in fact, in many studies, the opposite turns out to be true. Things are found in the autopsy that more than justify the treatment of the individual. There can be a long time to generate a meaningful report. One of our jobs is to, once we've looked at the slides, we've looked at the specimens, we've looked at all of the evidence, we've put it together in an autopsy report. And that report can be full of jargon, and it doesn't necessarily answer the questions of interest for the clinicians. But now we allow them to ask specific questions of us that we try to address during the autopsy. The College of American Pathologists, which regulates how pathologists practice throughout the USA, has provided guidelines now, and these mandate that a report be produced within a reasonable time frame, even in complex cases. And we think it's extremely important to communicate the results of the autopsy to families and discuss the implications of the findings. So for example, is there evidence from the autopsy that if there's a neurodegenerative disease, that it might be a genetic form of the degenerative disease, which is important for the family to know, the children to know, the relatives to know. So we make a point of communicating the results to the families and discussing the implications of the findings and discussing the autopsy findings in lay terms, because we realize that the autopsy itself the autopsy report can be full of a lot of verbiage and jargon that most people don't understand. There are many benefits to non-forensic autopsies, meaning hospital or academic autopsies. It's very good education for practitioners and trainees. What's very important is that we identify and clarify emerging or re-emerging diseases. So when I came to UCLA, it was the beginning of the AIDS epidemic, and we were really seeing a new disease evolve before our eyes. And it was extremely illuminating to examine, for example, the brains from individuals with AIDS and neuro-AIDS. Legionnaire's disease at an earlier time was characterized by autopsy studies, some viral diseases like West Nile virus, and yes, of course, COVID has to be in the mix. We've done several autopsies on COVID-19 infected individuals, and there's a lot to be learned from those individuals as well, especially questions pertinent to their neurologic problems. There are local quality assurance aspects of autopsy practice, and autopsy data provide fairly accurate vital statistics as well, or contribute to vital statistics, and provide data on the quality of care. So if for example, Dr. Magaki's study, she studied many individuals who had been in nursing homes and was able to establish the contributing factors in the death in very elderly individuals, sometimes with or without neurologic diseases. We now know that many people are living to 100 years of age or older, and so we now not uncommonly see autopsies from centenarians or even super centenarians. I think the oldest individual on whom we've done an autopsy was 114 years old. I saw in the news yesterday that the great actress Olivia de Havilland died at the age of 104 in Paris. So people are living to an older age, and it's important for us to make sure that their life is as comfortable as possible during that time. But when they do die, to use neuropathologic tools to study brain tissue. I think I'll skip this slide actually. A number of myths about the autopsy, common misconceptions. One is that the autopsy is a disfiguring procedure. It's definitely not. I can assure you of that. It's feared that the autopsy may delay burial by days or weeks. That's not true. We do the autopsy as soon as we get the consent for the autopsy. And if individuals have a religious requirement, for example, that the body needs to be buried or cremated the day after death, we can accommodate to that as well. In some religions, the family requests the presence of a clergyman during the autopsy. We are perfectly fine to have that happen. And so all of these are not impediments to the autopsy. There's a myth that dead tissue is useless in providing helpful diagnostic information. That's not true. In fact, many viruses in the body can actually survive a few hours. In the 1980s, we actually provided tissue from the brain of an AIDS patient who had a unique neurologic syndrome. And one of our virologists here was able to characterize the virus from that individual. It was a strain of virus that is known to have a tropism for the brain. And then I mentioned previously the fear that the family or the attending MD won't get meaningful information in a timely fashion. But we always make sure we are available to discuss autopsy results with the family. So I'll say a few words about Parkinson's disease specifically. I realize this is the Parkinson's Movement Disorder Alliance. You know that Parkinson's disease is the second most common neurodegenerative disease after Alzheimer's disease. Other types of degenerative disease of the nervous system are less common. Frontotemporal lobar degeneration, we're not quite sure what the incidence is because it can actually masquerade as a psychiatric disorder in some individuals. There are common causes of Parkinsonism. The most common is idiopathic Parkinson's disease. Rarely, and this is really in the bailiwick of Dr. Subramanian and my clinical colleagues, a small number of Parkinson's disease cases are familial, resulting from genetic mutations, but most are sporadic. MSA is a multiple system atrophy, which is caused by the same misfolded protein as Parkinson's disease, but the distribution of the misfolded protein is quite a bit different than in Parkinson's disease. And then, of course, diffuse Lewy body disease, in which there is distribution of Lewy bodies throughout the cortex. And then I'm not going to talk about the uncommon causes of Parkinson's disease or the secondary or symptomatic Parkinsonism that results not necessarily from structural disease of the brain, but can be the result of toxins and various kinds. So this is again something that links Dr. Subramanian and me. We both went to medical school in Toronto, and when I was in medical school, but not when, well, he might have still been there when Indu was in medical school, a very important discovery was made by a Dr. Ole Hornikevich, who was actually of Ukrainian origin, but worked in the Brain Research Institute in Vienna. And he in early 1960, published a paper that showed the dopamine deficiency that was the key to Parkinson's disease. Now, this paper was published in German. In the 1900s, it was still common for high-profile papers to be published in a language other than English. I think that has pretty well changed now, so that virtually any important finding in science is generally communicated through the English language, and many journals that used to publish papers in other languages like German or Japanese have now switched to English. Dr. Hornikevich actually gave us lectures, or gave us a lecture on Parkinson's disease and his discovery of the dopamine deficiency. He's very interesting. He died just a few weeks ago in May of this year, well into his 90s, and he divided his academic time between Vienna and Toronto in Canada. So it was amazing to have this person who'd made such a fundamental discovery in Parkinson's disease give us lectures. Many people think he probably deserved the Nobel Prize for his discovery, but never received it, although he received many, many other awards for his disease. Now you're saying, when is this pathologist finally going to show us a pathologic specimen? And I will show you several. So this is a view of the midbrain, or the mesencephalon at the time of the brain cutting from a patient with Parkinson's disease. And this is the same structure, the midbrain, from a control without Parkinson's disease. And you can see the chief abnormality is that there is loss of pigmented neurons from the substantia nigra. Now you can't see that. I'll show you the microscopic confirmation of that. But the substantia nigra here has lost its black pigmentation. That black pigmentation results from a chemical that is in the neurons called neuromelanin. And neuromelanin is a breakdown product of dopamine. So when there's a dopamine deficiency, neuromelanin will not form, and the result will be Parkinson's disease. This is a higher power view. This is substantia nigra from a person without Parkinson's disease. This is somebody with Parkinson's disease. And you can see there is loss of neurons in the substantia nigra. And microscopically, when we look at those structures, this again is a control. I'm a big believer in showing control slides so you can see what the normal looks like. These are normally pigmented neurons containing neuromelanin in the substantia nigra. And this picture shows about an 80%, 90% loss of pigmented neurons, neuromelanin containing neurons in the substantia nigra. So relatively normal, and about 80% loss of neurons in the substantia nigra. Now the substantia nigra is not the only structure in the brainstem that contains neuromelanin. There are others, and all of those structures are prone to develop the cellular pathology that I'm about to show you, which is the presence of Lewy bodies. The Lewy bodies were discovered many decades ago, and they're structures that we can recognize on an H&E stained section. This is a section of the substantia nigra from somebody with idiopathic Parkinson's disease. And these two little spherules in the cytoplasm are known as Lewy bodies. This is their characteristic appearance. And they certainly correlate with the presence of idiopathic Parkinson's disease. They can be found fairly widely distributed in the brain. And eventually they can make their way, they can form in the cerebral cortex, and that usually correlates with cognitive abnormalities and diffuse Lewy body disease. And these contain the misfolded protein alpha-synuclein. They contain other proteins as well, but this is just a higher power view showing you a larger Lewy body. They have this magenta color, this sort of a glassy appearance, which is quite typical, and they have a little halo, but they're easiest to see in neurons that contain some residual neuromelanin, and they can be really quite abundant within the brain. Now, how do these contribute to the causation of Parkinson's disease? We don't know entirely, but we know that they are a very strong correlate with Parkinson's disease. They can also occur in the cortex, and they can be a little bit tricky to see in the cortex. They look like little bubbles in the cytoplasm. Here they are, and there's actually 3 Lewy bodies in the cortex here. There are some other neurons that do not contain Lewy bodies. And this is another section of the cortex showing a nice glassy Lewy body within the cortical neuron. Now, we are very fortunate to have antibodies to these different misfolded proteins that help us to characterize neurodegenerative diseases in detail. And a very important discovery was made some years ago. One of the genes that is mutated to give you familial Parkinson's disease, and Indu may correct me if I'm misquoting anything, but is actually a gene for alpha-synuclein, which is a kind of a synaptic protein. That gene can be mutated to give familial Parkinson's disease. There are other genes that can give you that are mutated to give you Parkinson's disease. But somebody had the bright idea that if alpha-synuclein is mutated to give you familial Parkinson's disease, that might be a key protein in the lesions of Alzheimer's disease. So these very sharp individuals created good antibodies to alpha-synuclein. The people who first did this, I believe, were Virginia Lee and John Trojanowski at the University of Pennsylvania. And sure enough, when you made antibodies to that protein, you could stain a histologic section and localize those proteins in neuronal cell bodies. So this really revolutionized how we look at Parkinson's disease and how we can study Parkinson's disease using immunohistochemistry. Immunohistochemistry is just a tool that was developed in the 1970s and is really a mainstay of pathology now, where we have antibodies to a given protein, we can layer them on a tissue section, and we can link that to something that allows us to see the localization of that abnormal protein in the brain or elsewhere. So it's a really, really powerful tool. Here's a higher power view of Lewy bodies, alpha-synuclein immunoreactive Lewy bodies, in the cingulate gyrus, which is a gyrus within the brain, which is one of the common locations for Lewy bodies to localize in the case of diffuse Lewy body disease. And here's a higher power view showing this very, very strongly alpha-synuclein immunoreactive Lewy body in a cortical neuron in somebody with diffuse Lewy body disease. So let me close by saying a few words about the importance of the autopsy consent. We would never do an autopsy without an autopsy consent. The only autopsies that are done without a consent, of course, are at the medical examiner's office. In fact, one of our former residents is John Lucas, who's the chief coroner, chief medical examiner for the County of Los Angeles. And a very effective individual as well, very good forensic pathologist. But academic autopsies are only done with the appropriate autopsy consent. Many medical centers now insist on signed and witnessed copies of autopsy consents on the appropriate institutional form. So we're very strict about that. Telephonic consents have been discarded pretty well because we've gotten into some regulatory problems with those. The signature of the person giving permission for the autopsy must be appropriately witnessed. And there is a hierarchy of next of kin, beginning with the patient's spouse, and then siblings and so on and so forth, children, siblings, and so on and so forth. And we very strictly observe that hierarchy of the next of kin, meaning the autopsy consent has to be signed by the next of kin. If there is no next of kin, it can be signed by the executor of someone's estate in the absence of the next of kin. Again, I would emphasize the pathologist must observe restrictions on the autopsy, no matter how counterintuitive they may seem. For example, when I first came to UCLA, we had a restricted autopsy. We had an autopsy on somebody who had died of a malignant brain tumor. The one restriction on the autopsy was that we could not examine the brain. It seems terribly counterintuitive, but the family were okay to have the rest of the autopsy done, the rest of the body examined, but not the brain. We absolutely abided by that restriction on the autopsy. California is one of the states where ante-mortem consents are allowed, meaning that one can consent to his or her own autopsy, but we usually re-verify the decedent's wishes with the family or the next of kin, even if we have consent given for the autopsy while somebody was alive by that individual. But when I first came here, we performed an autopsy on somebody with multiple sclerosis who had consented to his autopsy about 20 years previously, and we proceeded on that. What are the questions to be answered by the autopsy? Very often, the autopsy, the clinician who requested the autopsy, or sometimes the family requests the autopsy, have specific questions that they would direct to the pathologist. And there is now a field in our autopsy consent saying, do you have specific questions that you want to direct to the pathologist? And we try to answer those. And those trying to address those questions can guide the autopsy protocol and any modifications that are required. So we might do special sampling and special stains to answer the questions in that autopsy. For example, right now we don't know how extensively the brain is involved in COVID-19. We've had, I think, 7 or 8 autopsies in COVID-19 subjects, and we have sampled those brains very extensively to try to get a sense of what abnormalities might be unique to the brain in those individuals, because as with AIDS in the early 1980s, we're looking at a new disease with a new spectrum of clinical and pathologic manifestations. And again, we want to provide meaningful feedback to the clinician requesting the autopsy. So if Dr. Subramanian, after looking after a patient for many years or possibly decades, might have very specific questions about the brain in that individual, and we will try to do our best to try to provide feedback to her that she can then in turn discuss with the family, or we're happy to discuss it with the family as well. Timing of the autopsy, because many of the diseases with misfolded proteins like Alzheimer's disease and Parkinson's disease, those proteins are very stable over time. So the time interval between the death of an individual and when we remove the brain is not absolutely crucial to getting valuable information. So we can get very important information even with what we call a prolonged postmortem interval. That's the time between the patient's death and when we do the autopsy. A short postmortem interval is optimal if somebody has a metabolic disease, in somebody where we plan to do molecular or biochemical studies, because although the tissue itself is stable, various molecular components of the tissue might degrade, like the DNA or the RNA, but it's getting easier and easier to retrieve those materials from the autopsy tissues. And if an infectious agent is suspected, it's good to do the autopsy as rapidly as possible after death. And as I mentioned to you, we've certainly cultured HIV from postmortem brain specimens on more than one occasion. I'll just finish off by saying a couple of words about the other neurodegenerative disease. This is the brain of somebody with Alzheimer's disease on the right. This is somebody with cognitively normal brain. And you can see the normal cortex, normal ventricles, normal lateral view of the brain. By contrast, here you can see tremendous shrinkage or atrophy of the brain, which we know occurs in Alzheimer's disease, and very much marked enlargement of the ventricles. And we know that Alzheimer's disease very often involves the hippocampus quite early on. And these are the structural landmarks of Alzheimer's disease. These are neurofibrillary tangles in the cortex, and we now have ligands for the protein in those tangles. But again, this is another protein misfolding disease in which one of the proteins that is misfolded is tau, and that leads to neuronal death. But we now have very specific antibodies, and those are being used now in sophisticated imaging techniques. And this is my final slide. This is for the brain of somebody with a non-Alzheimer dementia without Parkinson's disease, in which there is very selective atrophy of the temporal lobe caused by another protein folding abnormality, which I'm not going to go into at this time. So this is what we produce at the time of our brain cutting, and we try to answer the questions of the clinicians or the families. So I guess my final comment would be that autopsy is not a disfiguring or very complicated procedure, and it can give a huge amount of information. And it's really provided very important clues to further characterize pathophysiology. For example, Dr. Hornikavitz's paper in 1960 was really a landmark paper in understanding neurodegenerative disease. And I think it's about 1/4 and I'll stop with that and be happy to take any questions. I'm glad we didn't get black screen, so that was good.
Yay, that's awesome. Do you want to? Yep, I think that's perfect. That was very thorough, Harry. Really answered a lot of questions that I think I've had even as a clinician, thinking about, referring and how long you can wait. It sounds like you can wait a while, even if the body's been around for a while and things like that. So that's really helpful. One of the questions was about correlating imaging to autopsy. And I think we've been pretty lucky to have a group at UCLA that has, a lot of multidisciplinary folks that care about the patients a lot. And I know in brain cutting, sometimes we have the radiology team come and learn, and it's really been nice. Do you guys have a lot of correlations between imaging and pathology?
We do. The problem with many neurodegenerative diseases is the last imaging was done, a year, months or years before the death. The best correlations we have are in brain tumor patients, where patients have been imaged usually right until a few days before they passed. And there we definitely do it. And we actually will cut the brain the same way that the MRI cuts the brain, so that we can do side-by-side comparisons. Yeah, I should have mentioned that we really enjoy having our neuroradiology colleagues come to the conference. We enjoy our clinical colleagues coming, but also the neuroradiologists who are there. And actually our chief of neuroradiology here, as you know, Dr. Noriko Salomon, is actually a very good neuroanatomist and is a very astute neuropathologist. So she kind of keeps us on the straight and narrow and keeps us honest. It's really great having her there.
That's really interesting. So there's been a few questions just in the chat. So one of the questions is about the cost, just in general. What's your sense of the cost of an autopsy?
So there is if somebody has a UCLA medical record number, there is no cost for the autopsy. It's considered to be part of their continuing care. If the patient comes from an outside hospital, they've never had any association with UCLA, we will do the autopsy. My understanding is that the cost for a brain-only autopsy is $2,000 all in. That includes from gross examination to sectioning and interpretation. For a whole body autopsy, I believe it's $3,500 or $4,000, and that will include the brain. But if somebody has a medical record number, even if somebody was seen for an appendicitis 35 years ago, if they have a medical record number, that autopsy is done without any charge whatsoever, and our staff in the autopsy service are paid for out of the Global Medical Center budget.
There's a question here about how do autopsy numbers in the US compare to Canada and the EU? Are there similar trends in the US as there are in outside the US? So I guess, we saw that I was pretty struck by the figure that at Mass General Hospital looked like there was almost like 85% of the people that died there got an autopsy back in the 50s. Yes. And then, as we've sort of changed, I think that at that time we had very little neuroimaging, and really everything was sort of from autopsy driven to understand what was, really taking the lives of some of our patients. But now that we have imaging, I think it has probably decreased. But what's your sense of outside of the US?
In comparison to Canada actually, I think does fewer autopsies than we do. So as you know, I do some consulting work. I do locum work in Edmonton, which we talked about yesterday. And they do less than 100 autopsies a year there. We're going to do over 200 this year. In Europe, it used to be the practice that anyone who came into hospital pre-consented for their own autopsy. I don't think that's the case anymore. So I suspect the numbers are down there as well, but I think Canada probably does fewer autopsies than we do. We get a good autopsy population because many of the smaller community hospitals simply won't do autopsies. So they call us, transfer the body here, and we do the autopsy for them.
Is your sense, this is a very activist type group on the chat here, and people are watching this videos after, and there's really been a push to try to get, things changed in legislation and policy and stuff, throughout the world. So Harry, as somebody who does this type of research and relies on the brains to really do sort of help the cause along, what would be some things that patients can really lobby for? I think one, it sounds like Medicare used to pay for autopsies, and now we're relying on funding from other places, and it sounds like a lot of people aren't wanting to do them just because they don't get paid. So what are things that are real life, you have some patients here that you can teach?
Yeah, the really important thing is to delve into or lobby for well, research funding, but to emphasize that many translational research findings are dependent on human biospecimens, and those biospecimens usually come from autopsy. So, so that's one very important clue to get to politicians, or one very important pressure point for politicians. In our Alzheimer's Center, I give a talk every year on the value of brain banking and the autopsy, and how it's really revolutionized how we think of Alzheimer's disease as well. But we're getting to the point where various treatments of disease are going to modify structural changes in the brain. So, so the autopsy will be very important as a public health measure to show how effective a treatment has been or not been. And that's certainly the case in Alzheimer's disease right now.
One of the other things that's been interesting is that, on death certificates, when people pass, that things are listed on there. And then some people have a sense that, and I think this is real, that, when we say that people don't pass away from Parkinson's as the cause of death, that may detract from funding or media attention or lobbying for these diseases. What's your sense of that from the pathology community? And also, as somebody who knows maybe a little bit about this, what the coroners list and things like that. And is there some way that we can influence that when we're talking to our doctors, or if a loved one passes away? Is there a possibility to sort of influence what those are, what gets written down?
That's a very good point. No, you're absolutely correct. Death certificate data are wildly inaccurate, and they very often don't list the underlying cause of the disease. They just list the mechanism of the disease. So, for example, somebody with Alzheimer's disease might die of a pneumonia or pulmonary embolus. That would be the proximate cause of death, whereas Alzheimer's would be lower down on the list. I'm trying to think of what the solution to that would be, because very often those death certificates are filled out kind of in a hurry and somewhat superficially, and more accurate reporting of the cause of death would be valuable. So, for example, the figures that are now bandied about COVID-19 as a cause of death, I've heard that there are many inaccuracies in those death certificates because they list COVID-19 as the proximate cause of death, even when there was some other thing that was overwhelming in that individual. So they're extremely inaccurate. And when I hear, I could give specific examples of misstatements that I think I've heard from public health officials about that.
Yeah, that is a problem, I think, because my experience is that people have basically been, whoever fills out the death certificate is sometimes just the person who happens to be at the hospital. So,
exactly. Yeah.
So I think that could be a place where we can align, put some energy that could change outcomes, I think, of funding and things. And also I think that possibly pushing for more autopsies and pushing for more funding for autopsies in the system would be helpful as well. There's been a question or two about the GI findings in Parkinson's. What are you finding in those when you do cut the brains, and maybe your colleague had done that study?
We haven't looked at certainly there are reports of alpha-synuclein in the GI tract ganglion cells. The problem is most of our Parkinson's disease are brain-only autopsies. So that kind of gets to the issue I mentioned, that it's more helpful to have a complete autopsy, especially since we're realizing that Parkinson's disease or synucleinopathies in general are very widespread. And then there's a question of, does it move? We were talking yesterday again about the movement of these abnormal proteins from ganglion cells in the GI tract to the brainstem, and then thence to the cerebral cortex. So we haven't got a lot of experience looking at the GI tract, but there certainly are reports of alpha-synuclein present in the GI tract.
So it sounds like if you guys had the ability, you would want to try to get autopsies on every patient with these neurological diseases, if possible, whole body. It sounds like it would be helpful to have patients come to centers where they're examined pre-mortem, of course, like in life, by
Yeah.
doctors and document things, and then be able to have a sense of what maybe imaging looks like, what a sense of what the clinical picture looks like, response to medications, and then to do the autopsy. So you have a full picture really to be able to guide research and things like that.
That would be extremely valuable. And you know, we very often, people who are doing translational research studies where they expect patients will be coming in from outside will write into their budget for the study costs of autopsy based on anticipated deaths in that study. And that facilitates the autopsy as well.
It also sounds, there's been some questions just about where to document. So even if people have things documented, you know, maybe in their will or in their advanced care plans, sort of they've talked to people generally about this idea, it sounds like unless they fill out a form to donate the brain formally, this is so in life. And
Yes.
then it sounds like they have to tell their loved one that they would want that so that the loved one can sign off. And that consent has to be done in person or seen by a loved one at the time of death in order to, a witness signature by a loved one then is also required. So there's a few different steps to decide, to communicate to a loved one, to fill out a form before you die, and then after having that loved one be able to sign off in order to get this whole process sort of squared away. Is that accurate?
Yes, that's fairly accurate. We actually, for our Alzheimer's Center, we keep a binder of pre-mortem consents or ante-mortem consents, either from the next of kin or the patient themselves in the morgue. And when our Alzheimer's Center was at its peak performance, we would have a body come in, we would check that binder, it's alphabetically organized, and we would proceed with the autopsy. It was usually given by the next of kin or the patient, his or herself. We maybe might have lost you.
I think maybe Dr. Subramanian's kids.
One
I
of
think
my
they're
kids is
musicians.
yeah, my musician. It's not a violin, it's a guitar. Sorry about that.
I thought it sounded great. He sounds talented.
Yeah,
Yeah,
sorry about that.
so we keep a binder with the pre-consents, either signed by the individual or more commonly the next of kin. If there's a long interval between the consent signing and the death, we will generally re-verify the consent.
That sounds good. Well, Harry, this has been really a pleasure to have you. I'm just going to give you a couple more minutes maybe to close out. Any sort of other final words from an activism standpoint or research standpoint that might be helpful for people as well. I think one of the other things, I know there's been a lot of specific questions about consents and, you know, this place versus that place and different institutions. I think what I will say is that each place has a very different set of policies. And so if people are interested in this, I urge you to reach out. I think Dr. Adler had come on and talked specifically about some of the Mayo Clinic policies. Really important to get a sense of this in your own regions. Plan in advance so that people aren't stressing out sort of at the last minute is important as well. I think what happens is that there's really a wish and people don't necessarily communicate that wish to their loved ones. And then people are scrambling and then it ends up that, you know, things don't come to pass. So I think it's really just so important to communicate a little bit, you know, before and have these important conversations. And I think you've given us a wealth of information, Harry. And if there's also any other resources that you might have that we can link in regarding this or neuropathology information, because I think one of the questions also is, you know, there's probably a lack of great neuropathologists. There's a lack of movement disorder neurologists and, you know, some of these subspecialty folks, really, there's just a shortage across the world really of these people who really know this field well. And so, you know, if there's any resources that you have of maybe lists of brain donation programs that you've worked with or consortiums that you know, all collaborate with or something like that. If there's a central place that people can go to look for this, I think it would be really helpful. But I'll just give you a minute or two to kind of close for us.
Yeah, I think activist pressure on politicians is very important. I mean, the classic example of activism really moving a research field forward was in the early days of HIV research, where HIV research, even though it was a huge public health problem, was really funded out of proportion to its frequency in the population. And that was because otherwise young, healthy individuals were speaking to politicians, lobbying politicians to get funding for that. The same can certainly happen with families of individuals with neurodegenerative diseases. Even if the patient who has the disease is disabled or doesn't feel they have the energy or ability to lobby, they can certainly convey that enthusiasm for lobbying to their families, younger members, and they can then exert political pressure to increase the funding and increase research in those areas. Very, very important theme. But yeah, the classic example of activism moving a field forward was really HIV, and it went from being a deadly disease when we did our first autopsies. Almost inevitably, the patients died in a severely emaciated state in 1982, 1983. By 1996, it was basically a chronic disease that is totally manageable. Now, admittedly, that's a viral disease, and antiviral agents work quite effectively. The protein misfolding diseases are a little bit different, but activism is always extremely important. And I will try to get some contact links, some information to you.
Well, thank you so much, Harry. As always, you're always such a pleasure to see, and I'm glad we could connect yesterday as well in person. And I think that what you're seeing, hopefully, from this series is that we just have some really amazingly brilliant minds working behind the scenes. Sometimes these unspoken heroes that we don't really get to, you know, sort of see or highlight. And I wanted to bring one of them, who is Harry, and Harry's shown you his entire team that works behind the scenes and really, you know, connects these dots and, you know, works to get people their reports and the final answers that they may have been, you know, waiting to learn about. So I think that communication, the collaboration, the sort of camaraderie, I think is so important. And I think we, the patients, you know, people who care about our patients, people living with Parkinson's and their loved ones, and the advocacy organizations are part of the fight that needs to happen to get, you know, these dollars and the sort of brains and really the tissue that is going to take this disease and make things happen, you know, in the future to really change the outcome. So I think HIV is a good sort of example of really people working together to really kind of, you know, revolutionize something. So I think that the energy is there. So let's work together to kind of make this happen now. So thanks again, Harry, for
my pleasure.
time. And I'll hand it back to you guys, and it's for our final goodbye.
Thank you, Dr. Subramanian. Thank you so much, Dr. Vinters, for joining us today. That was fantastic. Before we do our wave off to everybody, I just want to let you know Dr. Subramanian will be back on Wednesday on the future of DBS. And we also have a really great talk tomorrow on why Parkinson's symptoms might abruptly change. So if you have time and can join us, we look forward to seeing you again. And until then, let's do our wave goodbye. Thank you so much for joining, everybody. We will see you again soon. Take care.